So hopefully everyone's well rested from lunch, feeling very eager. So we're very happy to have Kaie Kubjas here to tell us about genome reconstruction.
Thank you, Martin, and thank you for the organizers. Thank you to the organizers for inviting me. I'm going to talk about joint work with Sean Dewar from Bristol, Georg Grasegger from Leeds, Fatemeh Mohammadi from Leuven, and Anthony Nixon from Lancaster. And I was hoping that the paper will be on archive by this talk, but it's not yet, but soon. I would like to start with advertising, and I know that it was advertised earlier this week, but so the deadline to submit your paper to Mega is next week, but there's a tradition to have a extension of a week, so if you're still considering. So the conference is at the end of July, beginning of August in Leipzig, so please submit your papers. And if you have any questions, please talk to Frank or Simon or me about the conference. So I'm going to talk about how to reconstruct 3D genome organization from a matrix that we will call, or what is a high C matrix. And I will focus on single cell setting. So this means this matrix here, although you can't see it right now, it's a 0-1 matrix. So single cell means that it's a matrix coming from one cell. Depending how I go with the time, I might tell also a little bit about the population setting when we have a matrix coming from a collection of cells. Right, and so maybe a little bit more. So what is this? Or I will tell about this matrix more in a couple of slides. But so you probably are all familiar that the ordering of nucleotides is important, or what nucleotides we have, but maybe have heard less about the three-dimensional structure. So how the chromosomes are like aligned in the three-dimensional space, and so this is what this talk is about. And so there has been research that also the three-dimensional organization of the DNA plays an important role for gene regulation. So it's only not the sequence of nucleotides, but this together with how the chromosomes are placed in the space. So we have this 0-1 matrix that's called a single cell high C, or single cell contact matrix. And it indicates whether there's a contact between genomic loci. So how you should think about this, you have your chromosomes, and they are partitioned into segments. Let's say one segment is 100,000 nucleotides. And then and you can think that the rows and columns of this matrix, they correspond to this genomic loci. So let's say 100,000 nucleotides. And then 1 in this matrix indicates that there is a contact between the corresponding genomic loci, and 0 says that there is no contact. Okay, so I will what this contact exactly means, so we will have different interpretations, and I will get back to this. But you can think of this as contact means that two genomic loci, they come close together in the three-dimensional space. And 0 means that they are further apart. Okay, and so this is our starting point. So this is something that can be measured, so using what's called chromosome conformation capture techniques. So let's say 10 years ago, it was mostly for this population data, so that you had collection of cells, and the outcome was a matrix that wasn't then 0-1 matrix, but rather a matrix of positive integers for this collection of cells. In the last years, single cell data has been available. So that's our input. and we would like to construct a three-dimensional structure or say something about the three-dimensional structure based on this 0-1 matrix. Okay, so we model the DNA as a string of n beads, and the n beads, they correspond to this genomic loci. So we had the chromosomes, and we partitioned them into different loci, and each of the beads corresponds to one locus. Okay, so you can think of this, it's some kind of discretization of the, or aggregation of the genomic information. And we will record the positions of the beads by, in a matrix X with rows x1 to xn. And so the x1 to xn, they are points in Rd, and so we are primarily interested in the real life setting when d is equal to 3, but actually many of the results that I'm presenting will be also for d equals 1 and 2, just for the reason that these are the base cases, and actually often we can't, so we don't have a full understanding about the three-dimensional setting. So the main questions that we are studying is existence and uniqueness. So existence asks, so given a high C matrix, so this 0-1 matrix, and possibly also some pairwise distances between beads, is there a 3D genome structure with a given high C matrix and distances? Okay. There needs to be more information, like because what does it mean? There is a 3D genome structure with a given high C matrix. I have to tell you how these two things correspond. But that's the first main question, and the second one is when we add the uniqueness. Does there so if there exists a structure, is it unique, or are there finitely many, infinitely many structures? And then the second, then more practical question is how to reconstruct, so given a 0-1 matrix, how to reconstruct the three-dimensional structure. And we will study these questions across various models. So in this talk, I will tell about 3 different models. And it turns out that these models, they correspond to known frameworks in the rigidity theory. And so in our work, so maybe I shouldn't say this, but let's say that theoretical, so it's more building a dictionary between the what's happening on the biology world, and then trying to find out or like figure out what are the like notions from rigidity theory and results from rigidity theory that then translate to something that we're interested in on the biology side. So let me start with a quick introduction to the rigidity theory. So the main object in rigidity theory is a bar joint framework. So it's a pair G and ρ. G is a finite simple graph, and ρ is a realization of the graph. So it's a map from the vertex set to Rd. And the question that one asks is, what is the set of vectors or like realizations ρ prime such that 2 frameworks, G and ρ and G and ρ prime, have the same edge lengths? Okay. So first, this set is always infinite because ρ prime might be obtainable from ρ by a composition of Euclidean isometries. So if you apply translations, rotations, and reflections, then we get a different realization. So we will always work up to this Euclidean isometry. So we are interested, after discarding these trivial options, are there still infinitely many realizations with property that the frameworks have the same edge lengths. So there are three main situations. So if up to like Euclidean isometries, there are infinitely many frameworks, so G-raw, that have the same edge length, then we say that our framework is flexible. And here you can see an example of a flexible framework. For example, you could take the top vertices and then push them in and pull the like side vertices out. And this is how you get infinitely many frameworks that have the same edge lengths. If not, if there are only finitely many frameworks with the same edge lengths, then we say that the framework is d-rigid, or sometimes locally d-rigid. So d is referring to the dimension where we are considering our graph. And furthermore, if there are no other frameworks at all with the same edge lengths, then we say that the framework is globally rigid. Okay, and here you can see an example. So this is a locally too rigid framework, but it's not globally too rigid. Why is that? So can you see another realization of the graph? Exactly. So you could move this vertex, reflect along this line over here, and then you would get a different realization of the graph. But then, sorry, here it would have been maybe more instructive to do the same figure, but now add another edge between these two vertices, and then it becomes globally rigid, because then adding this edge, after adding this edge, we can't flip the vertex anymore.
Is it true that every graph is flexible for d sufficiently large?
I want to say, so I might be wrong about the rigidity theory questions, but complete graphs, I think they are not rigid for like no matter what d you use.
I mean, you take a triangle on the plane. That will be, so it'll be a triangle in some plane wherever you put it, and it'll be rigid in that sense. I mean, take a tetrahedron.
Yeah, it's a complete.
It'll span a free space. And
yeah, so complete graphs. Any other questions? Okay, so then I said there were these two questions, existence and uniqueness and reconstruction algorithm, and we study them for 3 different models. Actually in our paper for 4, but I'm going to talk only about 3 here. And so the first model is something that we call the threshold model. And at least to me, this is the most intuitive, and this is also something that we found in the biology literature. So in the threshold model, there is a contact between 2 loci if and only if their distance is below some threshold value dc. So I'm writing dc not to confuse with the dimension d. Okay. So, and to me this is very natural. So basically you put a ball around your bead, and so if there's other beads that are in this ball with radius dc, then you have a contact between them, and otherwise you don't have a contact between them. And furthermore, like we will assume that dc is equal to 1 because we can consider everything up to scaling. So, and it turns out, so as I said, these things that we're interested on the biology side, often there is already something notion that we want on the mathematics side. So for the threshold model, we consider something that's called unit ball graph. So a graph, G, is a unit ball graph if there exists a realization of the graph such that the distance between two vertices V and W is less or equal than 1 if and only if there is an edge between the vertices. So, and here you can see a graph that's a unit ball graph, but this is a unit ball realization of the graph, and this is not. So the balls are unfortunately not so, you can't see them very well, but basically at every vertex, there is a ball centered at that vertex, and then all the edges that go out, so the other endpoints of the edges, they should be inside the ball that's centered at that vertex. Okay, and that's not the case over here. So if you look, these two vertices, there's an edge between them, but their distances are greater than 1. Okay? So it is a unit ball graph because you can see a unit ball realization over here, but this is not the unit ball realization of the unit ball graph. And in the threshold model, there exists a 3D genome structure with a given high C matrix if the high C matrix is equal to the adjacency matrix of a unit ball graph. And also so if the dimension is 1, then the unit ball graphs, they're called unit integral graphs, and I believe also maybe indifference graphs. And if dimension is 2, then we call a unit ball graph a unit disk graph. Or we don't call it, this is what's called in the literature. Unit interval graphs, they are quite well understood. So if you have a unit interval graph, then G is chordal. So this means that every induced cycle in the graph has a chord, and every connected component of the clique graph of G is a path. So clique graphs are graphs where you have a vertex for every maximal clique, and there's an edge if the cliques are adjacent. Okay, and there is this characterization about the unit interval graphs. Unit disk graphs are already not so well understood. So there is this paper from 2018 from Discrete and Computational Geometry by Atminas and Samarayev. So they say that they show that these 6 are forbidden subgraphs in unit disk graphs, and there are also 2 infinite dimensional families in there, but it's not known that this would be a complete family of minimal forbidden subgraphs. And then already in dimension 3, that's the dimension that we would be interested in. Basically, we didn't find anything in the literature, so a small lemma is that the complete bipartite graph 113 is a minimal forbidden subgraph of the family of unit ball graphs in dimension 3. And the reason for this is that the Kissing number in dimension 3 is 12. So this means if you have a unit ball, you can have like at most 12 unit balls touching the central unit ball. But this is all that we know in 3 dimensions. And yeah, we expect that there are a large number of other minimal forbidden subgraphs, probably some that come from extending the minimal forbidden subgraphs from 2 dimensions. and possibly more. But the bad news is, so I like this threshold model, right? Because this is like very simple, very natural from the biological perspective. But then, so I said there is the existence and there is the uniqueness question. So existence we don't understand very well in 3 dimensions. And for uniqueness, there is basically bad news. Given a unit d-ball graph G, there are infinitely many unit d-ball realizations of G modulo isometries and scalings, right? So this is saying if you have a 0-1 matrix, and let's say we can figure out that this is an adjacency matrix of a unit ball graph, then there are still infinitely many realizations. And the argument is very simple. You take your unit ball realization. If none of the edge lengths is 1, so all of them are smaller than 1, then you can just perturb any of the vertices. If some of the edge lengths are 1, then you first scale by something just a bit smaller than 1, and then you perturb the vertices. Okay, so a simple model, but unfortunately no hope for uniqueness at all. Okay, and so this leads us to search for additional constraints. Yes.
Could you hope that they're up to like some notion of isotopy, where you, like you say, where you just perturb things a little bit? Isotopy is where you have some object in space and you're able to move it a little bit, like to form an oval or something.
Maybe we haven't. So what I'm going to go into is we are rather like saying, okay, now let's fix some edge lengths. So we haven't looked into that direction, but maybe this is something interesting to consider. Yeah, so we were looking for additional constraints that give finite number or unique reconstructions. And then the second model that we gave the name is threshold and microscopy. So in the unipole graph model, so this was characterized by a specific set of distance inequalities for edges and non-edges. And now we allow more general distance inequalities and equalities defined by distance constraints. So basically we have inequalities, but we also have like some edge length or like distance constraints for vertices. And 2 situations where this setting might happen is first when we can estimate the distances between neighboring beads. And I've talked to Caroline Uhler, who says this is a reasonable assumption to do. And the other thing is there is more and more imaging data available, and then estimating certain distances using imaging data. And the situation is better now when it comes to unique realizations. So let X consisting of x1 to xn be a generic set of points in Rd that satisfies a set of distance constraints and distance inequalities between pairs of points. Then further suppose that no distance inequality is satisfied as an equality. Okay, so if our distance inequalities are of this form, then we require that these points satisfy them strictly. Then there are finitely many sets of points satisfying these distance constraints and strict distance inequalities if and only if there are finitely many sets of points satisfying these distance constraints. Okay, so this is now a long proposition, but what it's saying is that inequalities by themselves can't get us from infinitely many configurations to finitely many configurations. So for this, we need the distance equality constraints. While inequalities alone do not determine whether there are a finite number of reconstructions, they can help to get us from finitely many to 1 reconstruction. Okay, so equalities are needed to get finitely many reconstructions, and then inequalities can further get us down to one reconstruction. Then whether the equalities define so whether there are finitely many configurations that satisfy the distance equalities, this is actually now the original rigidity question from rigidity theory. Okay, because we have a graph and we know some edge lengths, and we want to know how many realizations we have. Okay, and so I'm going to give a couple of classical results from rigidity theory. So one object that plays a central role is something called rigidity matrix. So this is, first rigidity map before we go to the rigidity matrix. So the rigidity map goes, so this is the, from the realizations of the vertices to the vectors given by the edge lengths. So we have a realization, and for every edge, it maps to the squared distance between the corresponding vertices, or the squared edge length. And then the rigidity matrix is the Jacobian of the rigidity map. And whether a set of distance constraints characterizes a finite set of points can be checked with high probability by looking, or generically by looking at the rank of the rigidity matrix. So there is this classical result from Asimov and Roth almost 50 years ago that says, let G be a graph on n vertices. A generic framework is D rigid if and only if either so the number of vertices is less or equal than the dimension and G is complete, or n is greater or equal than D plus 1, and the rank of the rigidity matrix is given by this value. Right, so this I already said, so special cases when we have only equality constraints. So when we don't have inequalities at all, so when we don't have the high C data, but we have the, let's say, data from microscopy experiments. And in this case, the uniqueness of a solution is equivalent to global rigidity. And so one of the results for about global rigidity of the graphs, again, from 1992 from Hendrickson. So let G be a graph on at least D plus 2 vertices. If G is globally D rigid, then we have this inequality for the number of edges in terms of the vertices and dimension. And there are also more recently a necessary and sufficient condition using stress matrices that, but I don't want to state it here. Okay, so that was the second model. Now we consider a third model that's related to Benny graphs. Maybe you have heard from other settings. So we consider a simplification of the unique ball graph model. Again, because the unique ball graph model, there we didn't have the unique realization, where contacts are defined by pairwise distances of 1, and non-contacts are characterized by distances greater than 1. So this model is more restrictive than the unique ball model. However, it offers the advantage of producing unique reconstructions in certain cases. And also it's a special case of the previous model. So basically we have some equality constraints for edges, and we have some inequality constraints for non-edges. Okay, so let me give the definition. So a D-sphere graph is the contact graph of a collection of unit D-dimensional spheres with non-overlapping interiors. Or in other words, there exists a realization rho such that when we have an edge between V and W, then the distance between V and W is 1 in the realization, and if there is no edge between V and W, then the distance in the realization is strictly greater than 1. And such a realization is said to be the sphere graph realization of G. And when we are in 2 dimensions, we use the term Penny graph. So this is very classical, and we made up the term marble graph for the 3-dimensional case. Okay. And so here you can see an example of a Penny graph with a Penny graph realization. So there are 4 pennies, and there is an edge. So there is 1 vertex for every center of a penny, and there is an edge between 2 vertices if the corresponding pennies touch. And none of the pennies can be overlapping. And this also implies that all the non-edges the distances corresponding to non-edges are strictly greater than 1. So, and here you can see another Penny graph, but not the Penny realization, because the distance between these 2 vertices is less than 1, because the pennies are overlapping. So what's known about Penny graphs on n vertices? So first there is an upper bound on the number of edges that you can see here. So this lemma is not ours, it's everything is from the literature. Then the minimum degree of a Penny graph is at most 3, and the maximum degree is at most 6. So the minimum degree comes from that there needs to be a vertex who's like that is convex, and then it can have at most 3 neighbors. And the 6 comes again from the Gissing number, but now in 2 dimensions. A maximal clique has at most 3 vertices. It's because at most 3 points can be equidistant on the plane. And then the chromatic number is at most 4. There is a similar result for marble graphs. So now we have marbles in 3-dimensional space. Then there is this bound for the number of edges, and it's not known whether the bound is tight. In the 2-dimensional case, the bound on the number of edges was tight. The minimum degree is at most 8, and the maximum degree is at most 12. The clique number is at most 4, and the maximum possible chromatic number is not known, but it's known to be between 5 and 9. Yes?
What kind of consideration gives that edge bound?
I don't know, I have to say. So this is a classical, or like, this comes from the literature. I can give you the reference, but I can't give you the answer. Sorry. And then the minimal forbidden subgraphs for marble graphs are K113 and K26. So that's what's known. That was about the realizations. Now about unique realizability. There is the notion, so let G be a d-sphere graph. We say that G is d-sphere rigid if there exist finitely many d-sphere realizations modular isometries, and we say that G is globally d-sphere rigid if there exists exactly one d-sphere graph realization. So this over here is d-sphere rigid, and these are d-sphere flexible, because you can move around the pennies. And if you have a d-sphere graph, then if G is globally d-sphere rigid, then it's also d-sphere rigid. And if G is d-sphere rigid, d is lesser or equal than 3, and the number of vertices greater or equal than d plus 1, then G is d-connected. So that was about the theoretical results about existence and uniqueness. And then, as I said, the other aspect that one is interested in is reconstructing the three-dimensional structure. So we propose a semi-definite optimization-based reconstruction algorithm. So for this, let me introduce some notation. So let G be the Gram matrix corresponding to the beats x1 to, or the points x1 to xn. So this means so G is n times n matrix, whose ij entry is the dot product of xi and xj. It is a positive semi-definite matrix of rank at most d, and it is related to the squared distance matrix. So a squared distance matrix for the set of points can be obtained from G. So the ijth entry of d squared is equal to gii plus gjj minus 2gij. Okay, so there is on one hand the Gram matrix, positive semi-definite, and then the distance matrix. And we will introduce this function, small gijg, that's precisely this linear form in the entries of the Gram matrix. Okay, and we use the following notation. So this is just notation that we have some set S equality. This is all the distances are like equality constraints for distances, and similarly we have non-strict and strict inequality constraints for distances. So for example, in the unit ball graph model, this set would be empty because we don't have any equality constraints for distances. Then the lesser equal constraints would be precisely for all the edges. So this would be lesser equal than 1. And then all the strictly, this set would consist all the pairs of vertices except the edges and repeated, so pairs corresponding to vertices. This is the program. So this is our program that we consider. So first, for the equality constraints, we have the objective function. So we want to so recall this gij. They corresponded to the pairwise distances, right? So we want to minimize the squared squares of squared pairwise differences between pairwise distances, subject to the inequality constraints that we have in our model. Okay, and then this constraints that the sum of gij's is equal to 0. here we want to get rid of the translations, and furthermore, the gram matrix need to be positive semi-definite. So this is where the last constraint comes from. So we use this, and that's the first step of the reconstruction algorithm. So when we run that program, then we get the gram matrix G hat, but we still need to get the points. So we find the largest eigenvalues of the gram matrix G hat and the corresponding eigenvectors, and we define the points xi using so this formula over here. And if G hat is a rank D matrix, then we so if you start if G hat is a rank D matrix, then it actually is a gram matrix of points in D-dimensional space, and we recover these points. But in general, if the rank is higher, then it won't be a gram matrix of a set of points in D-dimensional space, and then we do so. This corresponds to a projection to D-dimensional space. And this method is called classical multidimensional scaling. So we first do the semi-definite program, then we do the classical multidimensional scaling, and finally we improve the solution using gradient descent, starting with the solution that we found using the multidimensional scaling. And I'll show you some reconstruction. So the blue, you can think of the blue figure here as the our synthetic chromosome, so that's the same in all these figures. And then the red one is the reconstructed one. And we have used the different models that I introduced. So here is the unit ball, here is the penny graph, and the lower row is when we have the, all the inequalities coming from the unit ball model, but furthermore, we know some distances between pairs of vertices. So here we know for 2%, 5%, 10% of pairs of vertices. And you can see for 10%, one already gets a pretty good reconstruction. Okay? And so this was the unit ball model. So the reconstruction is, I would say, not so good, or it's quite off, but this is also in accordance with our theoretical results, that there is no unique reconstruction, right? So there is no hope that we could reconstruct exactly the structure that we started with. So we also then did comparison. So procrustes distance. So if you have two sets of points that are ordered, so procrustes distance is like is finding after transforming. So so it finds it minimizes the pairwise distances between the ordered sets of points. Okay? And you can see that for 10%, so the performance of the different models. So in general, besides, right, and the R here corresponds to the threshold value that we used for saying whether there is a contact or not. The penny graph model performs the worst. And then when we have at least some information about the pairwise distances, we get better reconstructions. Okay.
So that's the maximum, or the minimum of the maximum?
That's the
and distances. Is the minimum of that or the maximum of that?
It's the minimum. So you're finding the transformation that gives the minimum value of the. I don't think there is any max involved. Okay. So you're looking for the transformation where the sum of squares of so differences. Okay. Yeah.
Is there any intuition as to why I know that the R is equals 4 is somehow better than 5?
I'm thinking, so here in the synthetic experiments, what we did, so we used a Brownian motion model where, so between the neighbor, so we started with E, then we constructed the next one, and the distance between neighboring beats was always 1. And then we used the R to construct our Heisey matrix to decide whether there is a contact or not. And probably there is like some reasons, let's say if you analyze the distribution y r equals 4, maybe it's better than R5 or R6, but yeah, so this is what I can say about this experiment. And maybe regarding these R's, in the biology papers, so let's say roughly what is 1 is the distance between neighboring beats, and often in the experiments it said like it's around 2 or 3 is what is the threshold for saying that there is a contact. And also what so this Procrustes distance we can use only for synthetic experiments, because for this we need to know the original structure, because we are like aligning the original structure and reconstructed structure. So if we have real data, we can't use the Procrustes distance. We have something else called our violation function that measures the violation from the, so then we started a Heisey matrix, and we can we started Heisey matrix, we reconstruct, we can find the Heisey matrix for the reconstructed structure, and then we can use this for deciding how good was our reconstruction. And so we do also that, and what is actually surprising, the 5% of equations in that case performs worse than 2, so there is like some peak from 2 to 5 and then going down and then it goes down. So these two, let's say, ways to measure the goodness of our reconstruction give different results. Right, so, and the reason why the paper is not ready is because, so it's a math biology paper, and it's always desirable to have also experiments on real data. And so we have been doing these experiments for some time, but there are always like unexpected obstacles that come up. And you know, one of the problems is the data set that we're working. So the number of nucleotides is in millions, even if we take 1 chromosome, and then we need to somehow, our semi-definite program won't run on this data set, right? And so we need to do some aggregation, but then some of the results don't always, yeah. So there are always some unknown obstacles, as Oscar, who is in the audience and with whom we worked on a different 3D genome reconstruction paper, knows very well. But to conclude, so I talked about these first 3 models. So the threshold model that corresponds to a unique-ball model in rigidity theory. Then I considered threshold together with some distances. So on the math side, unique-ball and some distances. And then the same distance model that is then Penny, marble, or the sphere model. And then they correspond to, so in the unique-ball model, we have just inequalities for edges. In the second model, inequalities and equalities. In the Penny graph model, only equalities, and then non-edges, only inequalities or equalities. And there is this, you know, maybe this model is the simplest and makes most sense, but then there are problems with the unique reconstruction. And then in these more restrictive models, we get better theoretical results, but yeah, so the information in the second case, maybe the information might not be available. And in the Penny graph model, is it realistic that we assume that all the edges have the same length? So it has its own problems. So this was all about the haploid organisms, where we had one set of chromosomes. So it would be interesting to study single-cell genome reconstruction for diploid organisms, so for example humans, where we have 2 copies of chromosomes, and that introduces like more difficulties. So that would be something interesting to explore. And I think this is a good time to stop. Thank you very much for listening.
Questions?
Anna?
So you said about the MDS, the multidimensional scaling step, and then the gradient descent step. So in the gradient descent step, you're also incorporating distance information. Is that right?
So in the gradient descent, what we do, so we try different versions. I believe in the current version, basically we're incorporating only the high C matrix information. So when we have one, then we want, so we penalize if the corresponding distance is greater than 1, and when there is a 0, we penalize when the distance is less than 1. But it's a non-linear objective function. We also tried with like adding the distance information. I don't remember right now why we decided to take it out, but I believe in the version that we use, we just use, so we have 2 different kind of terms in the objective function.
I see. And how does that usually change the solution, that step?
It improves a little bit, but not too much. So I feel like it doesn't hurt to do it because it's a quick step, like, but it's not like very significant change. Yeah. We haven't done, let's say, numerical comparison, like how does one could compare Procrustes distance, for example, before the gradient descent step and after. So maybe that's a good thing actually to do, like also some analysis, how does it change? Yeah. There was another question?
I wanted to ask what the vertices and the vectors are actually supposed to model. Like, in the beginning this was there, and then I got lost in the graphs and all the exciting geometry, but like what is actually living at every vertex in the data?
Mm-hmm. So let's say in these experiments, so you would have originally million beads or like million nucleotides, and let's say 1 bead is corresponding now to 100,000 nucleotides. You could think that this is like the mean of their positions. So let's say, although it's a string of beads, it's really the 1 point that we want that represents this 100,000 nucleotides.
And is it true that there's a scale where if 2 beads are like a uniform scale, where if 2 beads are actually connected, then the distance between the centers is less than that magic threshold value, and if they're not connected, then it's bigger than that? Like, this whole analysis seems to be predicated on some number that separates connected from not connected. Is that a realistic? I mean, I don't know anything.
So my very simplified understanding what happens, and Oscar can also help me with the biology part, is you have your cell. You add some chemical to the cell nucleus, and when parts of the genome come close together, then this chemical like physically glues them together, right? And this happens, when they're close together enough. Yeah.
I mean, I think it pushes them close together. I don't know if it glues them together because the distances. There's always distance, right? It depends on the types of bonds between the molecules. Right? I would assume that that's why you need a threshold in the first place, because there is no such thing as really zero distance.
Well, there is no. I mean, what happens? Okay, so there is somehow if 2 parts are close together, then so they're like there is a bond, or like what I said, clued. So of course it won't be zero distance. And then the nucleotides in the two parts, they're actually like read as a one sequence that are like in both parts. So I say cluing. I don't know what's the, let's say, more correct word, but then this sequence is read, and this is how like the two parts of the genome, there's a contact, because then like this read sequence is mapped to the two parts of the genome.
Okay, so one for my clarification, there is a difference between red and the actual physical nature of who reads those, right? So this is when you're observing. If you had the data on the genome, you'd be observing these distances being below some threshold, and then that's what you'd be marking. It is okay.
Thank you.
So, yeah, what do you, I guess, hope to get from like? Do you have a sense of what biologists want to learn from this sort of reconstruction, or?
So, I mean, one thing, for example, what I've seen in the biology papers, they discover, for example, there is some like peak loops in some regions, or like, for example, there is somehow you have a chromosome, and there is like something that measures bipartite index. So you have like one part of chromosome is very condensed, and then there is like less condensed, and then there is another part that is condensed, and this kind of properties about the genome, like that something happens. Yeah.
There's actually the proteins that you would, like the chemicals you would add is spread things out. You have globule domains and things like that, where things are clustered together. And there's different pieces of the genome that are active and not active. They do this to spread them out so you can access them, right? Part of the chromatin.
How does what you're doing compare to, is there, I don't know, completely different methods to do the same thing, and how does it compare?
So I would say now imaging has, like in the last couple of years, probably that's the future. But let's say in the last 10 or 15 years, the reconstruction from the high-C data has been very like many people have worked on this, but not so much from the identifiability or uniqueness perspective, but the focus has been on the reconstruction algorithms. So distance-based algorithms, probability-based algorithms. So like a little bit difference in our work is we have been interested in these uniqueness questions. Basically, like all these algorithms, like there was no question, like is there one structure? Like what do the algorithms reconstruct? But yeah, so there is an overview paper, and I think that overview paper has 30 different algorithms for genome reconstruction from high-C data. The paper is so I think the overview paper is probably a couple of years old now. So it's probably mostly for the population data, where the high-C matrix is from a collection of cells rather than from single cell. But yeah, I mean, I'm not an expert on this at all, but I think at some point it's, the microscopy data is, are getting so good that one doesn't have to do high-C anymore. But I think right now that's probably still the main, like let's say, the cheaper and more accessible option.
Any other questions? All right, shall we thank the speakers?